talk. All right. So Sergio Serrato-Arroyo is currently a PhD candidate at Arizona State University. And Sergio began his academic career at California State University in San Marcos. And after obtaining his BS in 2020, Sergio joined the lab of Dr. Sylvie Huijben at Arizona State University. And at ASU, he studies the factors that contribute to the evolution of drug resistance in order to prevent or slow down the evolution of resistance in the human malaria parasite, Plasmodium falciparum. And with that, I will let Sergio present his talk today entitled Investigating the Factors Contributing to the Emergence of Antimalarial Drug Resistance.
All right, let me share my screen. Thank you for that wonderful introduction. So you guys can see this now, right?
Yep, looks great.
All right. So yes, today I'll be talking a little bit about antimalarial drug resistance and going into some of the factors that may be contributing to this. Um, okay, so just to start, I'll provide you with a little outline of how my talk's going to go. First off, I'll give you a little bit of background on malaria and the problem associated with this disease. And then I'll go into my aim 1, 2, and 3 of my dissertation. And then we'll do a summary and a recap. And then we'll have time for questions at the end. So to start, what is malaria? Well, this is a disease caused by the Plasmodium parasite. And this parasite is an obligate one, meaning it can only survive and reproduce within a host and cannot do so without one. And this specific parasite is transmitted through the bites of a female Anopheles mosquito during a blood meal. And some common symptoms you would experience are very flu-like in the sense that you'll experience fever, chills, muscle fatigue, vomiting, and in severe cases, anemia. Now, there are 5 species of the Plasmodium parasite. However, the deadliest form is Plasmodium falciparum. So for the sake of this talk, we'll focus on this specific species. So this disease is prominent all across the globe. However, it disproportionately impacts tropical and subtropical areas. And in 2022, 1.9 billion people were at risk of contracting this disease. And out of that, over 200 million people were infected and over 600,000 were killed. And out of the deaths, over 70% of these deaths were attributed to children under the age of 5. So as you can see, this disease has a wide distribution, is very infectious, and particularly deadly to children of young age. Now, there are ways we can treat this specific infection, and this is through the use of antimalarials, which are a class of drug that can either prevent the infection or treat an existing one. Now, there are various antimalarials out there broken up based on their mechanism of action. There's different classes such as quinolines, antifolates, and the latest and greatest tool, the artemisinin. Now, the major problem we have is that resistance has emerged to every drug on the market. Now, if we take a look at this timeline here, if we look at the implementation of chloroquine in the late 1940s, resistance occurred within the first decade of its use. And as you can see, with the constant implementation of every drug, resistance has emerged. Now, of more concern is that resistance has emerged to our frontline therapy known as artemisinin combination therapies, and this happened in the early 2000s. And now this is not widespread. However, if it were to become widespread, we can see massive increases in malaria mortality and morbidity due to us not having an alternative drug to replace this current frontline treatment method. Now, as you can see with the subsequent implementation of every drug, resistance has emerged, which demonstrates that no drug is evolution proof. So as you can see, all our efforts are going into developing drugs, but not really understanding what factors contribute to the emergence and spread of resistance. So uncovering these factors can improve our malaria eradication efforts by allowing us to develop novel resistant management strategies that aim to prevent or slow down the emergence of resistance to our therapies. And through this, we can extend the useful therapeutic lifespan of current and future pharmaceuticals with the hopes of reducing the overall malaria burden and eventual eradication of this disease. Now, also, while areas start to near malaria elimination, understanding these factors will allow us to prevent the emergence of new resistance hotspots. Now, all of this brings me to my driving research question of what are the mechanisms promoting the evolution of resistance, and can we utilize this information to prevent or slow down the evolution of drug resistance in Plasmodium falciparum? So I'll plan on answering this question through three aims, with my first being an extensive literature review trying to understand the differences in malaria epidemiology that contribute to the primary emergence of resistance in Southeast Asia for sub-Saharan Africa. My next aim is to investigate the impact initial parasite density has on the growth dynamics of malaria parasites. And lastly, this is the aim that the R.C. Lewinson Career Award actually helped fund, and this has to do with elucidating the role that drug dose has on the competition between drug-resistant and susceptible malaria parasites. So the first, like I said, is a literature review going through the differences in malaria epidemiology between Southeast Asia and sub-Saharan Africa. And the reason why is that these two regions present major problems for malaria elimination in the sense that if you look at sub-Saharan Africa, it harbors a disproportionate bulk of the malaria load in the sense that over 90% of cases and deaths occur in this region. Now, if you look at Southeast Asia, more specifically the greater Mekong subregion, now it might play a smaller role in the malaria burden with only 2% of cases and 1% of deaths occurring there. However, historically, resistance has emerged in Southeast Asia and spread to sub-Saharan Africa. And we saw this with two drugs known as chloroquine and sulfadoxine pyrimethamine, with resistance both emerging here along this Thai-Cambodian border and spreading all the way to Africa, reducing the effectiveness of these drugs and leading to an increase in mortality and morbidity. Also, we saw methylquine resistance emerge here first, and also artemisinin resistance in the late 2000s. So as you can see, this disease, although it plays a minor role in the overall malaria burden, it plays a major role in the problems we associated with this disease, which begs the question of why does resistance primarily emerge in the low transmission region of Southeast Asia instead of sub-Saharan Africa? So I have undertook writing a literature review that addresses multiple hypotheses that try to explain this abnormal resistance phenomena that we observe. Now, within this review, there are 8 different hypotheses that span 3 main categories involving transmission intensity, life history, and societal and cultural differences between these regions. Now, for the sake of this talk, I can't go through all of them. However, I will give you a brief overview of each of these categories along with the hypotheses, followed by going into a deeper look or giving you a deeper look into transmission intensity and its impact on within host competition. So to start for category 1, this has to do with transmission intensity and how it may play a role in the emergence of resistance. In this particular part, I'm focusing on sub-Saharan Africa in the sense that this is known as a high transmission region, and meaning that more people will be infected and experience greater numbers of infectious bites, which leads to more multi-genotypic infections or infections that harbor genetically diverse strains. And this right here can lead to greater within host competition between parasite strains for the limited host resources such as red blood cells. And also due to more frequent malaria exposure, there's an increase in immunity. And also since there's more circulating parasites in the overall population, there is a better or more effective recombination rate that allows for the breaking up of resistance mutations. Now, all of this is important in the sense that it can reduce the overall emergence of resistant parasites compared to Southeast Asia, where we see lower transmission, lower multi-genotypic infections, low immunity, and lower genetic diversity. And so now we look at category 2, which goes into the evolutionary history with malaria and how that may play a role in the emergence of resistance. So it is hypothesized that malaria spilled over into human reservoirs from gorillas, and basically once humans started migrating out of Africa, they took those parasites with them, and along that, parasites had to encounter different vector species. Now this plays a huge role in the sense that if you look at sub-Saharan Africa, the major vector is Anopheles gambiae, while in Southeast Asia, it is Anopheles dirus. And it is thought that the mosquito species Anopheles styrus, when resistant parasites are infecting them, maybe they experience a reduced fitness cost, which allows them to survive in the mosquito and transmit compared to resistant parasites infecting the Anopheles gambiae mosquito in sub-Saharan Africa. It is also thought the genetic background of the parasites differ between these regions, which allows Southeast Asian parasites to have a greater propensity to acquiring resistance mutations compared to parasites in sub-Saharan Africa. It is also thought that human host factors differ between these regions. And for an example, there are specific blood disorders that are more prevalent in sub-Saharan Africa versus Southeast Asia. And it's thought that these blood disorders may basically play a role in allowing the resistant parasites to proliferate, let's say in Southeast Asian human hosts compared to sub-Saharan African ones. And now on to the final category that I'll be addressing in this review. This goes into societal and cultural perceptions on malaria and treatment practices that may lead to resistance in Southeast Asia. So a main point in this section is that people believe that falsified and substandard drugs plague the antimalarial market in Southeast Asia. And these drugs basically contain either little or no active ingredient, which will end up exposing parasites to suboptimal concentrations of the drug, which allow them to build tolerance and ultimate resistance at greater rates than they would in sub-Saharan Africa. Also, we go into whether individuals buy from the private or public sector in these specific regions. And it is known that that private sectors actually contain greater prevalence of falsified and substandard drugs. So maybe if Southeast Asian individuals are buying from the private sector more than public, they will be exposed and taking more of these drugs than individuals in sub-Saharan Africa. And also, it is thought that education on malaria and specific treatments may differ between these regions, which may make individuals in Southeast Asia more susceptible to self-treating and also taking monotherapy, which is not advised by the WHO. And also, they might lack knowledge on which treatments to take or effective treatments to take to actually cure the infection and even know whether they have malaria or not. So taken together, all these three different categories are proposing various different hypotheses that try to explain this abnormal resistance phenomenon. And like I said, I'm going to take a deeper look into category one, more specifically hypothesis one, which goes into lower transmission regions experiencing lower levels of within-host competition and whether or not that promotes the emergence and spread of resistant parasites. So to give a little bit more background is that most malaria infections are polyclonal, meaning they contain genetically distinct strains. And this is known as multiplicity of infection. So as we can see, sub-Saharan African infections are more genotypically diverse than Southeast Asian infections, with the multiplicity of infection being at least 5 parasite strains or more in Africa versus 2 or less in Southeast Asia. Now, this plays an important role in the emergence and spread of resistance in the sense that these resistant mutants can become suppressed in these multi-genotypic infections because they suffer a fitness cost, which usually is visualized through a decreased growth weight. And also in the event that it's a de novo emergence of a mutant, they emerge at low frequencies, which already set them up to be at a disadvantage in being outcompeted by the more susceptible counterpart. And so in this image right here, I tried to depict what this hypothesis is trying to say in the sense that if you look at panel A, this is trying to depict a low transmission environment where an infection has an MOI of 2. So as you can see, if you have only 2 strains, meaning you have a susceptible and a resistant one, these resistant parasites can acquire more resources due to there being less within-host competition, which can allow them to reach more transmissible densities and be transmitted upon the next bite from an affected female Anopheles mosquito. And then we have B, which is the high transmission environment, to where we have an MOI of 5, where we have 4 susceptibles and 1 resistant. And this can lead to greater suppression of resistant densities, which don't allow them to reach those transmissible ones, which then basically doesn't allow the subsequent spread of resistant parasites. So, like I said, this is a literature review. So I tried to scour, the literature to find um, evidence and support or against this hypothesis. And what I did find is that there was ample in vitro and in vivo evidence of competitive suppression. However, I only found one field study investigating competition's effect on drug-resistant densities, and this was in Africa. And basically, in this study, they took patients that were presenting malaria symptoms and then genotyped whether they were either resistant or fully susceptible infections and how many distinct parasite strains there were inside that specific infection. And what they found is that solo and mixed infections contain similar total parasite densities, which is indicative of uh, these parasites competing for the same amount of resources. They also found that the multiplicity of infection had no effect on total parasite densities. And the most important finding to me that they found was that resistant parasite densities were lower in mixed for solo infections. So as you can see here in panel A, this is looking at just chloroquine sensitive parasites. Yes, they were suppressed in multi-genotypic infections. However, when you look at the resistant one, they also were suppressed to lower densities than the susceptible one, which means that they possibly have a lower transmission potential. And so some preliminary conclusions I can make based off of scouring the literature for evidence on this specific hypothesis is that in general, there's just a lack of studies investigating within host competition's impact on drug resistance. Because there was only one study that investigated this in sub-Saharan Africa. However, the results from this study cannot be generalizable because they only looked at 3 different countries on the entire African continent. So we don't know if what we're seeing is just specific to these specific regions or it also occurs in Africa as a whole. Also, they only looked at chloroquine-resistant parasites, so we don't know whether resistant parasites to other, let's say, antimalarials experience the same competitive dynamics we observed in this one study. And they also did not investigate MOI's impact on chloroquine-resistant parasites in single versus mixed infections across sites. They looked at how it impacted total densities, but not whether if a resistant parasite was more suppressed in a infection with, let's say, 5 clones versus 1. They also did not investigate transmission potential of chloroquine-resistant parasites in single versus mixed infections. And also a major gap is that no field studies have investigated within host competition's effect on drug resistance in Southeast Asia. So what I think we should do going on in the future, future studies should investigate the effect MOI has on resistant parasite densities in Southeast Asia to see if we observe the competitive dynamics we observe in sub-Saharan Africa, to see if they happen in Southeast Asia as well. Also to uncover the transmission potential of resistant parasites in mixed infections, to see whether these parasites are basically investing more in transmission or asexual growth. And also, I think we should repeat studies like this one that I had just mentioned for parasites resistant to other antimalarials to see if the competitive dynamics we observe in this one study for chloroquine are generalizable amongst other resistance mechanisms. So now I am done talking about that. We're going to go into my aim 2, which goes into investigating the impact initial parasite density has on the growth dynamics of malaria parasites. So just to give you some frame of reference is that Plasmodium falciparum has 2 distinct life stages. It has the sexual one that occurs in the mosquito and the asexual that occurs in the human host. And this asexual phase is responsible for morbidity and mortality caused to the host, which is also known as virulence. And just to give you some insight into the life cycle, when an individual gets bit by an infected mosquito, they inject these infectious forms known as sporozoites that travel to the liver and would take at least 10 days to mature, to where they produce these merozoites that then burst out of the liver cells and then infect circulating red blood cells or nearby circulating red blood cells. And this virulence, just to bring it back, is known to differ between strains because these parasites have to balance between replication and survival. And how these parasites do this is through environmental stimuli. And there's stimuli that include nutrients, blood type, and parasite density, which impacts parasites' growth within the entire host. However, I want to really focus on parasite density because we know parasites regulate their growth rate at high densities acutely. So in the exponential phase when they're growing, we see that parasites start to slow down and alter their growth rate, as in the sense that they sense that they're too high or maybe that they're running out of resources. However, we don't know how these parasites regulate their growth in chronic infections and infections that last a very long time. So what I wanted to do was I wanted to see if this regulatory response was, I want to see the impact initial parasite density has on on this regulatory response in chronic infections. So how I decided to answer this question was I took 4 different parasite strains. I set up 4 different treatment groups with 5 replicates each, and I started parasites at an initial inoculum size of 0.1% parasitemia, or 0.1%, means 0.1% of the red blood cells are infected with malaria parasites. And then I also set it up at 0.2%, 0.4, and 0.8. And then what I did was I ran the experiment for 16 days or 8 replicatory cycles because the parasite takes 48 hours to complete one life cycle. And what I did was I took blood samples every 2 days. I extracted DNA from them and then quantified parasite densities using qPCR. So some results that I observed is that in the first strain, known as W2, this parasite exhibited a recovery phenotype at high densities. And what I mean by that is if you take a look at this graph, you see on the x-axis, here's days, the y is parasite density. And what we see in the purple and the blue treatment groups, which correlate with the 0.8 and 0.4, we saw that they peak, and then these parasites are regulating their growth rate by killing off some of them, and then they also hit this peak again. However, we don't really see this in the lower inoculum size group with the 0.2 and 0.1% hitting peak and then maintaining growth, which shows that these parasites have a different regulatory response depending on the initial inoculum size that they start off with. However, I wanted to take and then also I wanted to take a deeper look into peak parasite densities, because what I thought is that if you start off with higher, let's say 0.8%, you would reach a higher peak because maybe you would experience a lag time or a lag effect on this regulatory response compared between the groups. And as you can see, between the 0.1, 2, 4, and 8 group, there wasn't really a difference in peak parasite density reached, meaning that the carrying capacity is the same for all starting densities. However, I have not had time to look through statistical analysis yet or conduct them, so I don't know if these differences are significant or not. Same here, I wanted to look at absolute parasite density, which is looking at how many parasites were produced throughout the entire infection. And what I would see is that I would see more parasites being produced over the course of the infection in the 0.8% group compared to 0.1, because the 0.8 would start off at a higher density and be able to maintain that high density for longer periods of time. However, we can see we didn't really see that. Now on to the next strain, DD2. We saw that this strain exhibited a faster growth rate, meaning it peaked at a faster time in the 0.2, 4, and 8 group, rather than the 0.1% group that had an extended log phase, which could possibly be due to just the way we set up the experiment and the way the parasites just responded to that. However, a difference is that we only observed this recovery phenotype in the 0.8% treatment group, while the 0.2 and 0.4 exhibited this maintenance phenotype. So yet again, we can see that there is different dynamics we would observe between different initial densities, which shows that maybe the regulatory response is maybe greater in the 0.8% group compared to the other initial densities. Yet again, I wanted to look at peak parasite density, and as we can see, there wasn't really a difference between the treatment groups, which is indicative that the carrying capacity is yet again the same for all starting densities. However, when we looked at absolute parasite densities or parasites produced throughout the infection, we kind of saw the 0.4% group producing more parasites than the other treatment groups. However, we haven't run any statistical analysis, so we don't know if this difference is statistically significant or not. And so now for the last strain that I have data for, this is Hox. This is another parasite strain. And yet again, we're seeing some differences in growth dynamics in the sense that we don't observe the recovery phenotype in any of these specific treatment groups within this specific strain, which show that maybe this recovery phenotype is only observable in specific parasite strains. We do see that the 0.1 and 0.2% group take a lot longer to peak, which means they possibly just have a slower growth rate. And however, in the 0.4 and 0.8% group, we kind of saw that they peaked, maintained, and then dropped, which means that maybe the regulatory response in this strain is a lot different, or maybe the parasites can handle stress for a lot longer at higher densities. Yet again, we looked at peak density. We didn't really see any difference. However, what we saw in the absolute parasite densities was that something I had mentioned earlier was that what you would expect to see is that as the density increased, you would have a greater parasite production throughout the course of the infection, just because it stays higher a lot longer. However, like I said, we haven't run stats, so we don't know if this is significant or not. And so what I plan to do in the future is finish analyzing results from my last strain to see if we observe this recovery phenotype, or just to observe the growth dynamics in this strain to see if it's demonstrating things we've seen prior, or if it's demonstrating anything new. And also I want to look at the parasite multiplication rate, which is basically where we're looking at the doubling time, or the time it takes for a parasite population to double in time. And I would like to look at the max PMR that was experienced between the treatment groups, and also the peak PMR to see if we see differences in the parasite multiplication rate when the parasites are about to peak between these initial densities. So some preliminary conclusions I have come up with from this study is that it looks like inoculum size does not seem to impact peak parasite or absolute parasite density. Although we do observe different growth dynamics at various densities within strains, more particularly, we observe this recovery phenotype with higher density inoculations that we do not see in the lower density inoculum sizes. And this is also something, when I look into literature, I have not seen any other experiments observed. So we could be observing something very novel in the way we've set this experiment up. And also, since these experiments between the strains weren't set up at the same time, I can't statistically analyze between them, just because there could be confounding factors with, let's say, blood type, media we used. So, however, I visually observed between them, and we saw that there's differences or slight differences in growth dynamics between strains as well. So as we can see, initial parasite density does kind of impact this regulatory response and just parasite growth dynamics in general. And so now I'm going to go on to aim 3. And this is like the one I said where the R.C. Lewinson Award basically helped fund a little bit. I'm still working on this project, and we'll hopefully finish it in the summer. However, this one goes into elucidating the role that drug dose has on the competition between drug-resistant and susceptible malaria parasites. So just to give you a little bit more background, is that when you're infected with a mosquito, you're infected with multiple genotypes, and let's say you have a resistant one you're infected with, this one will become competitively suppressed in the presence of a susceptible competitor as the infection progresses. However, upon drug treatment, you will see that these susceptible parasites are killed off, leaving only the resistant ones to be able to survive and propagate to higher transmissible densities that end up leading them getting picked up by a mosquito and transmitted to another individual upon the next blood meal. Now, it is these principles of competitive suppression and competitive release that are thought to either promote or suppress the emergence and spread of resistant parasites. Now, we know drug treatment can select for resistance. However, there's various components of drug treatment that can actually do this, and we don't really know which components provide the greatest selection pressure. So what I wanted to do is I wanted to focus on one, and this is specifically dose. And I wanted to investigate the effects dose has on resistant parasite selection. And so how I plan on setting up this experiment is I will take a sensitive and a resistant parasite strain. I will grow them and synchronize them so they all start off at the same exact parasite life stage. I will set up solo and mixed infections. Then I will expose them to either a low or a high dose of drug, and I will also have a no drug treatment control to where every 2 days I will take blood samples and extract DNA from them and then quantify them using qPCR. And so the antimalarial of choice I have chosen for this is chloroquine. And just to give you some insight on what chloroquine is, this is known as a blood schizontocide, meaning that it prevents the parasite from maturing through the ring, trophozoite, all the way up to the schizont point. So the parasites end up dying and not really being able to proliferate. And how this specific antimalarial does this is that when the parasite enters the red blood cell, it utilizes hemoglobin and digests it for essential nutrients that it needs. However, a byproduct of digesting hemoglobin is this toxic form of heme, which then the parasite ends up changing to this inert, non-toxic form known as hemozoin. However, chloroquine comes to play is that once it gets into the digestive vacuole of the parasite, it binds to heme and stops the parasite from transforming this toxic form to a non-toxic form, which then leads to this toxic buildup of heme and ultimate death of the actual parasite. Now, there are parasites that are resistant to this specific drug, and this resistant mechanism is known to be as a metabolic type of resistance, meaning that when chloroquine gets into the cell, this specific Plasmodium falciparum resistance transporter effluxes chloroquine out of the cell, lessening its accumulation in the digestive vacuole and allowing the parasite to change heme into hemozoin. And this specific resistance mutation or resistance mechanism is mainly governed by the mutation at the 76 amino acid position in the Plasmodium falciparum chloroquine resistance transporter gene. And why I'm deciding to look at chloroquine and this specific PFCRT mutation is that these mutations are known to cause cross resistance between chloroquine and frontline treatment drugs such as imodiaquine. So as you can see, this utilizing this drug is still relevant because we need to observe how these mutations are altered by drugs and competition. And so what I plan on doing is taking 3 chloroquine sensitive and 3 chloroquine resistant parasite strains. I will set up solo and mixed infections of these specific strains. And these solo infections will start off at 0.05% parasitemia, while the mix will start off at 0.1% or 0.05% of each strain, so we can accurately compare what will happen in a solo flask versus how competition impacts parasite growth dynamics in the mixed cultures. And basically what I'm going to do is I'm going to set up 3 treatment arms with 5 replicates each. I will have a no drug treatment control. Then I'll have a treatment group that's exposed to a low dose, which is equivalent to an IC25 or an inhibitory concentration that inhibits the growth of 25% of the parasite population. And then I will also expose them to a high dose, which is equivalent to an IC75. And here's a schematic of how I plan on doing this, is I will run this experiment for 16 days with taking blood samples every 2 days and replenishing nutrients or media for the parasites every 2 days. And on day 6, which is right around the time the parasites peak or right before they peak, I will implement the drug chloroquine and basically implement the drug all the way until the conclusion of the experiment. And through that, on day 6, on day 8, I will change media, take blood samples, and also replenish the drug so the parasite gets nutrients and new formulations of the drug every 2 days. And just to give you insight on how I will get these IC values that I'll need for this experiment is I'll perform dose response curves. And basically the flow of this work is that I will take the drug chloroquine, I will make a stock solution, take malaria parasite culture, and I will create a series of serial dilutions to where the drug starts very concentrated and gets less concentrated throughout the serial dilution. I will expose the parasites to the specific drug for about 48 hours to where then I will take blood samples, extract DNA from those, and then quantify parasite numbers using qPCR. And then what I would do is I will basically compare parasite growth in the wells that were exposed to the drug versus the control to see what that specific drug dose did to the parasite population. And so some preliminary results I have from this is I have one dose response curve for the strain DD2. And if you take a look here on the x-axis, this is drug dose. The y-axis is inhibitory concentration. And as we can see, as the drug dose increases, more and more parasites die, with kind of the optimum of what I need to really like hone in on being between, let's say, like 80 to, let's say, 200-300 nanomolar. So like I said, this project is still in the works, and I hope to have it done by the end of this summer. And so to summarize and recap is that malaria is a deadly disease that kills hundreds of thousands of people every year. And the major problem we have is that anti-malaria resistance develops and threatens to impede malaria elimination efforts. And so instead of focusing all our efforts in developing new drugs, we need to start understanding what factors contribute to the emergence and spread of drug resistance. And doing this can help improve malaria elimination efforts by allowing us to recognize how these efforts impact malaria epidemiology. And through that knowledge, we can develop intervention strategies specific for low transmission regions, because we know resistance emerges in those regions more frequently than in high transmission regions. And with that knowledge, we can also prevent the emergence of new resistance hotspots. And so just some acknowledgements is that I'd like to thank my PI, Dr. Sylvie Hyben, for all her ongoing support throughout this entire PhD program, and for her always trying to push me outside of my comfort zone and grow as a scientist and an individual. And then the Hyben Lab, everybody, but more particularly Max, Yesenia, Sasha, and more recently, Dr. Joe Segovia, because these people are on the culturing side of the lab. You know, we share a lot of experiences, and a lot of them have played an integral role into helping me acquire some of the data I presented to you today. And also for the Society for the Study of Evolution for granting me the R.C. Lewinson Award, and also this opportunity to present my work to you all. And then friends and family for always offering support and guidance throughout this PhD, but also making Arizona feel like home. And then I'd like to thank you all for listening, and then we can have some time for questions.
All right, great talk, Sergio. Really liked it. It looks like there's some people clapping. Thanks for explaining an area that I didn't know much about so well. I understood each point as you went through it. Really enjoyed it. If anybody has questions, you can unmute or and put your video on, or you can type them in the chat and I can read them. But if not, I'll start. So I'll start it off. So I was wondering, so like the resistant malaria comes into the environment in Southeast Asia, and it's able to flourish a bit more because there's fewer strains co-infecting, right? But then this resistance is then causing problems in Africa, is that right?
Yeah, because so there was a study, let's say for chloroquine.
Yeah.
They found that they looked at like the resistance mechanism, right, or like where the mutations happened,
Mhm.
and they looked at like these like flanking regions outside of it. Uh, they looked at parasites in Africa and Southeast Asia, and they saw that those flanking sites were very similar, or they were similar, indicative of like, let's say, a selective sweep, meaning that these parasites made their way to Africa.
Mhm.
This could have been through, let's say, people, traveling to Southeast Asia, being infected and not noticing, and then going to Africa, and those parasites, ending up establishing in the African population.
Yeah, that was what my question was about. Like they're in Southeast Asia, they're capable of establishing because the co-infection rate is lower and they're able to better compete with the non-resistant strains that don't have the fitness disadvantage. Right.
Yeah.
But then they moved to Africa and now they're dealing with a co-infection rate of about 5, which usually uh, limits the resistant strains that would originate in Africa. So how is the Southeast Asian strain capable of penetrating the Southeast Asian resistant strain capable of penetrating into the African population in a way that the African resistant strains cannot?
You know, that's what we're trying to, figure out right now.
Okay.
It's a very complex system. As you can see, there's a lot of different factors that go into play. But what I'm thinking is that once those resistant parasites are established in that population,
Mm,
they can just spread rapidly because people are taking these drugs, and since they're already there, it's like bam, you take away the susceptible, and they can actually just thrive in that infection and start transmitting.
I see, I see. So
Yeah.
they reach a certain population level and they're being introduced in large amounts or something like that.
Yeah, because what this review is really, really trying to do is understand why it primarily emerges there.
Mm.
And there's like hypotheses saying that, or what we think is that it it emerges there faster. However, if it gets into a high transmission environment, it'll spread a lot faster due to those competitive dynamics that we observe.
I see, I see.
That makes
Okay.
sense?
Yes, that makes sense. Thank you.
Okay.
And I'll let Derek ask a question here.
Okay. I didn't have a question off the top of my head. Give me a second here and I'll think about it. Good talk. I enjoyed it a lot. Um, I'm curious when you talked about like these visual differences that you haven't statistically tested yet. Do you think that you're going to find differences in the growth rates at those different concentrations, or do you think that's just a visual artifact that will go away when you actually test, if these parasites are growing at different rates?
Hmm. I mean, I guess based off of this is like my understanding of like error bars is that since they're very overlapping, I wouldn't really see any statistical difference, meaning that, you know, peak parasite densities, absolute parasite densities won't differ. However, we do observe these different growth phenotypes that aren't being shown within that the peak and absolute density data.
Okay.
All right, I'll continue some questions here. Um, I was wondering the strains you used in the second aim of the experiment, were those African or Southeast Asian strains? And did you expect that to matter at all if it was different?
Some of them they kind of varied.
Mm,
Some of them were like from Southeast Asia, and some of them were from Africa. Uh, I didn't really expect to see those types of differences in growth because no, all the papers I've read, I haven't really seen like this recovery phenotype, you know, or this yeah the type of dynamics I observed because a lot of the time when these parasites basically peak, they crash out, they get too stressed, and there's not. They think it's because there's too much, let's say, metabolic waste being produced to where
mm.
it kills them. So I honestly did not expect to see any of this. Um, and also, I can't really say, let's say it is. Well, actually, no, I actually probably can't say that. Never mind. I'm gonna stop.
Okay. Yeah, just whatever you're sure of.
Yeah.
Um, and then I mean I have some more here, but I'll stop it. But how about this is the last one? I was wondering anything about the South American strains because it's in South America too, right?
Yeah.
How do those compare to Africa, Southeast Asia, like the differences between Southeast Asia and Africa with South America?
Like as in like resistance levels, or like what do you mean?
Yeah, like so for example, the African malaria are usually like co-infected more, and that's less so in Southeast Asia. Like, how is it in South America? Is that like a good breeding ground for resistance, or is it in the middle, or is it like not good?
Yeah. So specifically for South America, it does resemble more of like a Southeast Asia type environment. It's a low transmission region. However, falciparum is not really prominent in that area. It's another species known as vivax, and we
Okay.
haven't really seen major problems with resistance in vivax.
Oh, okay.
So that's why we're focusing mainly on Southeast Asia and Africa because falciparum is more prominent there. Vivax is also prominent in Southeast Asia, but falciparum is a basically a bigger problem there.
With the resistance and all that.
Yeah, and just yeah.
Mhm. Got you. Okay. Well, I don't see anything in the chat here. So I think we'll call it there. And really thanks Sergio for a great talk. Like I said, a lot of complex things to break down, and you did a great job. So yeah, we'll see everyone back here for next week's Greg seminar. Thanks everybody for coming. Appreciate it. And thank you,
Yeah.
Sergio, of course, for your presentation.
Yeah, thank you for giving me the opportunity. You all have a great day.
Absolutely, you too.